Hello and welcome everyone to today's Science Breaks talk. My name is Gunnar Prusner and I will be chairing today's event. Today's talk is entitled The Bloody Battle Against Leukemia, and it will be given by Professor Cristina Lo Celso, who is a member of staff and conducts her research at the Department of Life Sciences here at Imperial College London. Unlike me, Professor Lo Celso is an experimentalist, whereas I'm a theoretician at home in the Department of Mathematics. I will explain in a moment how I fit into the picture and how Professor Lo Celso and I ended up collaborating. In her talk, Professor Lo Celso will explain how she and her research group use advanced microscopy techniques to understand better how leukemia works, how it interacts with infections, and how medicine can help people to recover from it. Professor Lo Celso asks, how do individual diseased cells behave differently compared to healthy cells? How do infections alter the behavior of some cells? And how does the Haematopoietic system, the system inside our bodies that produces around 1 trillion cells every day, work reliably? For many people, this area of research has life-changing consequences. We all have heard of people or know of someone close to us suffering from leukemia. Professor Lo Celso has dedicated her work to winning the battle against cancer. Professor Lo Celso obtained her PhD in biochemistry during her studies at the Cancer Research UK London Research Institute in Fiona Watts' group. She went on to spend 3 years at the Harvard Medical School, first as a postdoc and then as an instructor. She joined Imperial in 2009 and was made a professor last year. She has won numerous national and international accolades, such as the Fawkes Medal Award, presenting the Carl Zeiss Lecture, and most recently receiving the Royal Microscopical Society's Life Science Medal. I would like to add a personal remark. Cristina for the first time when we both took a tour of the Francis Crick Institute 4 or 5 years ago, when it was still being built. I was happy to learn that Cristina was willing to explain her research to a complete neophyte like myself, and I was completely blown away by the sophistication of her experiments that she is undertaking in her lab. Cristina and I immediately realized that a theoretician like myself can help her in her research. You see, I characterize random motion of particles, and so it wasn't too far-fetched that my group would contribute to her research by characterizing and identifying the motion of cancerous cells. We published a paper together last year with her and mine PhD students as first and second authors. Cristina's talk today will touch on some of this research. Before handing over to her, I would like to cover a couple of housekeeping rules. Please submit questions to Professor Lo Celso at any time via the Q&A section on the side of your screen, or by posting a comment if you're watching on the live YouTube stream. The presentation will be recorded, and if you don't feel comfortable with your name being read out, please submit it anonymously. I expect very many questions to be asked, and Cristina may not have enough time to address all of them. To promote any questions that you find particularly important or interesting, you can like them in the Q&A section. I will try to make sure that Cristina has a chance to answer the most popular ones. So without further ado, I would like to hand over to Professor Lo Celso to talk about the bloody battle against leukemia.
Thank you, Gunnar. It is great to be here and talk about what we have been doing over the last couple of years, looking at how healthy and malignant hematopoiesis interact in the bone marrow. Let's see if I can move the slides. There we go. So what I'll do today is tell you a little bit about my favorite tissue, which is the bone marrow, encased deep inside bone, which contains the entire process of hematopoiesis. This starts from blood stem cells or hematopoietic stem cells, and you might hear me calling them HSCS during the talk. And they're in charge of maintaining the production of red blood cells, white blood cells, and platelets throughout our life. Now, This means leading to the production of billions of new cells every day throughout our lifetime. And for example, it has been calculated that we produce more blood cells than there are stars in the entirety of the galaxy. Now, how does this happen? It's not that blood stem cells just know it all and they're magic cells that can do it by themselves. They do this because they reside inside the bone marrow and they interact with many, many different cell types. A number of them are presented in the scheme here on the right. And you can see we have both stromal cells, for example, osteoblasts, which are the cells that make bone. And therefore, we find that the interface between bone marrow and bone, which we call endosteal surface. But we also have mesenchymal stromal cells. We have many vessels, which are delimited by endothelial cells and other types of perivascular cells. And we even have progeny of the blood stem cells contributing to regulating the stem cells themselves. For example, megakaryocytes or T cells or macrophages. Now, what we're interested in understanding, both myself and my research group, is what signals allow HSCs to do what they need to do, especially when the demand for their function changes for various reasons. So, for example, we know that when we have severe infection, and this is particularly topical these days as we're about to get into various degrees of new lockdowns, we know that hematopoietic stem cells are put in overdrive and they really need to act and proliferate more and generate extra progeny to generate the extra white blood cells that we need to fight the infection. And this is often at the expense of the production of red blood cells and platelets. We also know that, for example, as leukemia develops, leukemia is cancer of the blood, which typically develops within the bone marrow, and it grows occupying the bone marrow space. As it does so, it actually leads to literally an obliteration of the healthy hematopoietic system, to the point that some leukemia patients will end up in the clinic because they have symptoms due to the lack of healthy cells and not so much due to the presence of the malignant cells. Now, again, all these phenomena take place within the bone marrow, and we know that they have an effect on the composition of the bone marrow, what we call the bone marrow microenvironment, and we really like to understand what it is that is happening. Now, it is nice to think about it when we have a relatively simple scheme like the one we have here, but in reality, looking at the bone marrow has been more challenging. In this image at the bottom here, I'm presenting a preparation of a mouse femur, where you can see how many cells are inside the bone marrow and how packed they are. And you can appreciate some of the structures, for example, some of the vessels that run from left to right in the middle of the marrow. Now, not only the tissue is encased in bone, which has made it traditionally quite inaccessible for direct visualization, but it contains very many different cell types, and we know that the events that are taking place there are dynamic, and so the relationships between the different cell types change over time. So in order to study this, what has been very useful for me and my research group has been to utilize intravital microscopy, because that allows us to, first of all, combine multiple modalities of microscopy, but especially it allows us to look inside the bone marrow of anesthetized mice and see how cells are behaving over time and what is happening directly in situ and in real time. So in this image here, I'm showing you one example of the typical images that we generate. We're looking at the bone marrow contained in the frontal bones of the mouse. And you can see a couple of structures because we have engineered the situation so that we have fluorophores expressed by particular cells and structures. So in green, you can see the osteoblasts, which are the bone-making cells that I just mentioned. And you can see how they're organized in clusters, relatively evenly spaced along the bone marrow. And we can also see in blue vessels. In particular, we can see a large vessel, which is a large vein here in the middle. And on the two sides of it, you can see these much smaller vessels that are crisscrossing the space in this honeycomb structure. And these are the sinusoids or the capillaries that crisscross the entirety of the bone marrow. Now, this image here is a 2D projection of a much larger 3D image that results from stitching together a number of smaller images. And the reason why I'm telling you this is because what this allows us to do is to have both this type of bird's eye view of the entirety of the bone marrow cavity, but it also allows us to focus on areas of interest, and we can still have very good single cell resolution detail of information on these areas. For example, here we can start appreciating single osteoblasts and single capillaries. Now, with intravital microscopy, not only we can identify areas of interest, but we can then also follow them over time. So for example, this example here is the first one I'm showing you of leukemia cells, of a specific type of leukemia, which is called T-cell acute lymphoblastic leukemia. It is in red here. And what we learned from this type of movies was that the T-ALL cells are migratory, and they move within the bone marrow space. And you can see this cell has now divided, and we now have two T-ALL cells. Now, if we look at the mouse a little bit later, we now see not only these single cells, but we can see also little clusters of cells. And the interesting thing here is that even though we're starting to have cell clusters, the cells are still all very active, and they're moving around pretty much constantly. So if we wait long enough, eventually these growing cells are going to occupy the entirety of the bone marrow space, as shown here on the left. And now the interesting thing that we can do is that we can start testing the effect of multiple treatments on these mice. And we can follow the very same mouse before being treated and being highly infiltrated with leukemia. The same mouse was then treated with dexamethasone every day. And dexamethasone is one of the drugs that are commonly used for treating T-ALL. And what we can see in the image of the right is a clear example of how dexamethasone works really well to debulk the disease, because the vast majority of the red signal has been lost. But we can also see really what is the problem and the challenge in treating leukemia, which is that a number of leukemic cells survive treatments, and we can see them as single cells or small clusters in multiple areas across the image, and so across the bone marrow. Now, not only these cells are there and survive, but what we found out was that these cells are highly migratory. We can see on the left cells surviving dexamethasone in red, and on the right we are seeing cells that survive a much harsher treatment, which is a combination of dexamethasone, vincristine, and asparaginase, which is again used commonly in the clinic for treatment of T-ALL. If we compare the two movies, we can see that with a harsher treatment, we have fewer cells surviving. But the interesting thing is that the fewer surviving cells on the right are also migrating faster than the cells surviving a milder treatment on the left. And another interesting thing you can see here, this cell has just disappeared and will appear again in a minute. This cell is not migrating, and it is also disappearing. It is actually literally dying. And actually, in collaboration with Gunnar, we have seen how if we manage to reduce the speed of the migration of the T-ALL cells, this also correlates with a higher amount of death. We're still left with a number of questions, though. For example, we want to know why, after treatment, the migration speed of the cells increases, and also why there is anyway a heterogeneity and some cells are faster than others. Now, what happens instead if we do not treat T-ALL? What happens is that the leukemia keeps growing for another little bit. It entirely occupies the bone marrow space. But as you can see in this image here, you can see in red is the leukemia, in gray is the signal from the bone. And I have to tell you that we are still looking at one of these mice where we should see osteoblast in green. And you can see just about a couple surviving on the left. And unfortunately, the vast majority of the osteoblasts have been lost. And this happens at very late stages of disease. Now, we were able to look a little bit earlier than this stage, and again, we performed a time-lapse microscopy that allows us to see our green osteoblasts shrinking and breaking into small debris of cells. And this is a process that is highly reminiscent of apoptosis, which is one of the types of death that cells can undergo. And in fact, we were able to demonstrate that indeed osteoblasts are lost by apoptosis. Now, this time-lapse movie was acquired over the course of about 8 hours. And if you can imagine extending this over the course of about 48 hours, I think you can easily imagine how we could end up in a situation where all the osteoblasts are lost very rapidly. Now, for us, what was very interesting to test was whether this was the case only in mice or also in humans. And what we could see is that we could stain bone marrow samples for osteoblastic markers, which are in brown here, and we can see osteoblasts nicely lining the endosteal surface, where the bone is here at the top of the image, in a healthy bone marrow section. And we can see that in a bone marrow section from T-ALL patient, and we saw this in multiple patients, if we follow the endosteum, we no longer see our nice osteoblast that we just looked at a minute ago. Now, more recently, we started focusing on acute myeloid leukemia. And we did this because, unlike T-ALL, acute myeloid leukemia is particularly associated with causing loss of healthy hematopoiesis in the patients. When we started looking at this disease, we saw that a number of characteristics were in common with T-ALL, but of course, a number of them were also different. So first of all, in this bird eye view of the bone marrow in the frontal bone of the mice, again, we can see in green endothelial cells, which line the vessels, and in gray is the bone. And this time, the red cells are acute myeloid leukemia rather than T-ALL. You can see that also in this case, we end up with clusters of disease, clusters of cells that little by little grow and grow. The other thing that we noticed is that also AML cells are migratory, and we can see them here migrate within the bone marrow space. However, what we noticed is that if we treat them with a drug that does slow down the T-ALL cells, not much happens in the case of the AML cells. And what this told us was that it prompted us to perform further analysis that showed that, in fact, the molecular interactions that are exchanged between AML and the bone marrow microenvironment are more complex, and there are many other factors that the AML cells can rely on in order to survive within the bone marrow parenchyma. Now, one other thing that we noticed when we were performing our time-lapse images was that if we compare what we saw inside the vessels in the bone marrow of mice that either had tomatofluorescent protein expressing healthy cells on the left, or that they had their bone marrow fully infiltrated by tomatofluorescent protein expressing AML cells, we noticed that exclusively in the AML mice, we could notice every once in a while a green debris going by in the circulation. And these debris are highlighted by these little arrowheads that show up here and there. Now, this may not look like much if we are looking at it as a movie, but I think you will be convinced of the difference between the healthy and the leukemic mice when I show you a time projection of these movies. And now you can really see the myriad of small GFP-positive particles that we see uniquely in the highly infiltrated AML mice. Now, these small debris reminded us of the small GFP-positive debris from the dying osteoblast, and they prompted us to have a more in-depth analysis of what was happening to the vasculature in the mice that were developing AML. And to make a long story short, one of the things we found out is that AML reduces the number of vessels that run very close to the endosteum. So I'm showing you a couple of examples here from sections. We are looking at sections from healthy mice at the top and mice that are heavily infiltrated with AML at the bottom. And I'm not showing you AML at the bottom because otherwise we wouldn't see anything else, because it would occupy the entire field of view. But again, we can see in gray the bone, and you can see in yellow we have these little vessels that run directly adjacent to the endosteum. And these very vessels are entirely lost in the situation where we have high AML infiltration. This is very obvious also in the metaphyses of the bones, which are basically the extremity of the long bones, for example, the area close to the knee for the femur. These areas contain what we call trabecular bone, which is a honeycomb structure of bone that remodels very actively, and it's primarily in charge of supporting our weight. What we see is that this area is highly vascularized in the healthy situation, and these vessels, because there is so much bone and so much endosteum, they are essentially endosteal vessels, and we can see that they're highly reduced when we have high levels of AML. We also looked in human samples, and again, when we focused on the 20 microns closest to the endosteum, we could identify endothelial cells in sections from healthy bone marrow, but this area was pretty much devoid of endothelial cells in AML samples. Now, one of the reasons why we were interested in these vessels is because they have been associated to healthy bone and healthy osteoblasts, and also healthy hematopoietic stem cells. So we went back to our bird's eye view of the bone marrow, and what you can see in this image here is in red is healthy hematopoiesis, in yellow is leukemia, in cyan are our patches of osteoblasts, and then in blue we can see vasculature. So this is one of the images that we spend quite a lot of time looking at to really understand what is the interaction and how does the intermixing between healthy and malignant hematopoiesis takes place within the bone marrow. And what this clearly showed us is that we have these areas that are highly infiltrated of leukemia that almost looks like there is a core of leukemia cells that has an advancing front, that as it advances, it eliminates healthy hematopoietic cells, and we now know it eliminates also healthy stroma, and we can see in this image it eliminates healthy osteoblasts. We don't see clusters of osteoblasts where we have many YFP-positive leukemia cells. And if we focus now on this area here that is in the square, and we are just going to look at one slice, image, and we can see the bone as well, you can see how on the left, where we have healthy hematopoiesis, we have our nice patches of osteoblasts along the endosteum, and these are immediately interrupted once we reach an area that has dense leukemia infiltration. So then at this point, it may not come as so much as a surprise that when we looked for hematopoietic stem cells in the areas of bone marrow that would be highly endosteal versus not, and these are the metaphyses, which I presented to you earlier, and the central marrow, which we can isolate from the diaphysis of the long bones and separate from the endosteal area, we could see that as leukemia developed, while we didn't lose many hematopoietic stem cells from the middle of the marrow, where we are far from the bone, instead we had a very strong effect on and a very dramatic loss of hematopoietic stem cells in the metaphysis areas. Now the story became a lot better because other groups have been studying these endosteal vessels, and they have identified both pharmacological and genetic strategies that can be taken advantage of in order to potentiate the endosteal vessels. And so, for example, one of these involves treatment with a drug called Deferoxamine. And while the bad news was that the leukemia grew exactly the same, and again, I'm not showing it to you in these two images, however, we could see that when there was Deferoxamine treatment, we could see many more vessels running next to the endosteum in bone marrow section. And in this case, we were also able to count many more hematopoietic stem cells in the metaphysis area of the bone marrow. When we use the genetic rescue of endosteal vessels, the interesting thing that we saw here is that when we administered chemotherapy, we could see that now chemotherapy was more effective. Not only we had fewer cells, fewer leukemia cells surviving at the end of the chemotherapy here on the left, but also when then we left the mice for a longer period of time after the end of chemotherapy treatment, the mice that had more endosteal vessels, which are the ones in purple here, had less infiltration of AML for much longer, especially in the peripheral blood. So what this work told us was quite exciting, and it really demonstrated to us that targeting the bone marrow microenvironment or targeting the interaction between leukemia cells and the bone marrow microenvironment is promising if we want to develop novel therapeutic interventions for leukemia patients, and we are working more in further developing this. But we also had another question, which is what happens to the bone marrow microenvironment when we have severe infection, and can we potentially intervene there as well? So here I'm showing you some work that we have been doing looking at the effect of malaria, which is a disease caused by Plasmodium infection, which can have extremely severe consequences for the patients, and that also affects over 200 million people worldwide every year. As I told you at the beginning of this talk, we know that during severe infection, hematopoietic stem cells proliferate more, and it is the case in this experimental model as well. So what we did instead was to look, for example, at components of the bone marrow microenvironment, such as the osteoblasts. So here we are looking in green at the osteoblast of a healthy mouse, and we notice that by the time we are at the peak of infection, and this only takes 7 days, we are again in a situation where we have lost the vast majority of the osteoblasts. Now, what is interesting for us here is that the mechanisms of loss of osteoblasts are different in the 3 situations I have shown to you, the T-ALL, AML, and infection. Because in this case, osteoblasts are lost everywhere, evenly, little by little, and we just have rarer and rarer osteoblasts everywhere. However, the end point is the same, is that we end up with a bone marrow that is devoid of osteoblasts. The other interesting thing here is that in this situation, we have not lost hematopoietic stem cells. The stem cells are still there, and they're also more proliferative. But an interesting thing that we noticed is that if we analyze the transcriptome of these cells, and this is the expression pattern of thousands and thousands of genes in each stem and progenitor cells that we can isolate from the bone marrow of controlled and infected mice, we can see that in the control situation, where we have good osteoblasts, we can identify a good subpopulation of cells that scores highly for this HSC score, which refers to the expression of thousands of genes that have been identified to be associated with stem cell function. However, in the infected mice, even though we know that the stem cells are there, their transcriptional signature is really very much lost. So again, to make the story better, we started to think about ways of intervene on this and could we rescue the hematopoietic stem cells. So because we had loss of osteoblasts, we thought that we could try first by potentiating the osteoblasts themselves. And this can be done by administering parathyroid hormone for a number of weeks. And this was successful in rescuing osteoblasts, that we still had a significant amount of osteoblasts in green here at the bottom surviving following infection and the parathyroid treatment. And the hematopoietic stem cells also were there, and they were no longer pushed to proliferate heavily, like in the case of the untreated infected animals. So what we did next was to combine parathyroid hormone treatment with an antioxidant treatment, N-acetylcysteine. And in this case, we could again have hematopoietic stem cells that didn't proliferate much. Osteoblasts were partially rescued. And what was interesting for us to see was that if we compared the reconstitution potential of stem cells taken from healthy mice, infected mice, or mice that had been treated with our double treatment, if we can look at the green line here, this is a much better reconstitution than what we achieve if the stem cells are taken from the bone marrow of infected mice. They're not quite as functional as the healthy stem cells, which are in the black line here, but they're really much, much better than what we have at the peak of infection if we don't have any other intervention. So again, this is telling us that as a proof of principle, interventions can be engineered so that we can rescue the function of hematopoietic stem cells. And again, we're left with a number of questions. We know that leukemia can be treated and blood counts can recover. We know that infection eventually goes away, but what we still don't know is to what extent the stem cells and the bone marrow microenvironment recover from these injuries. And we think that if we can understand this, then we can then finally start understanding what are the links between infections and blood cancers, which are still very much beyond our comprehensions. And it seems that for certain cancers, infection can be a triggering factor, but for others, it may be the opposite. And I think that really only if we can understand the details of these interactions and these dynamic processes, we can then really get to the bottom of it and understand the mechanism driving it all, and ideally not only treat, but also prevent the development of blood cancer. So I will stop here, and I thank you for your attention. And I thought I would just very briefly mention the many funders that have allowed myself and my group to perform the work over the years.
Hi there, I'm really sorry, you're on mute. Sorry.
Oh, I'm so sorry. So thank you very much, Cristina, for this really interesting presentation. There's a solid number of questions in the question and answer section, and many have been liked. Thank you everyone who submitted a question. Before I put any question to Professor Lo Celso, I would like to apologize if I mispronounce anyone's name. So the very first question I would like to put to you, Cristina, is why is leukemia so common in children?
So leukemia is common in children or in the elderly. It has a very interesting biphasic incidence. And what we started to understand is that this very much depends on when the initial triggering mutations occur. And what we have learned is that in infant leukemia, these mutations happen already during embryonic and fetal development. This is a time when all the cells in the body are proliferating massively because a huge amount of growth is happening. And so because there is so much proliferation, there is in a way a higher chance that mutations may occur. And again, because there is so much growth, there is a higher chance that a mutant clone may grow favorably and take over the others and result in leukemia already in young children. If no mutations happen during embryonic and fetal development and they only happen in adult life, this is a time when hematopoietic stem cells are incredibly quiescent. So the chance of them acquiring a mutation is lower, and even if they do, then the time that it takes for the malignant clone to take over and really grow into leukemia, it is significant. And so this is why we have we have a peak of incidence of leukemia during the early years, and then again during the elderly.
Thank you very much for this. Maybe a follow-up question from Mike. How effective is targeted therapy in treating leukemia?
So it depends very much on the type of leukemia. For example, some leukemias are treated really well. An example is the promyelocytic leukemia, whereby if it is treated with chemotherapy and retinoic acids, because of the type of mutations that we have in this leukemia, we get a remission of 80–90%. When I say we, of course, the clinicians in the clinic and the patients. Another example that we thought was pretty much cured, and it turned out was a little bit more complicated, is the case of chronic myeloid leukemia, because this is driven by the BCR-ABL mutation. And years ago, drugs were developed that would target specifically that mutation. And as a result, it seems the patients were pretty much cured. What we are learning now is that new mutations still develop, and so eventually resistance can develop, and so it is more complex to treat the disease in that way. But generally, when we simply give chemotherapy, we don't always have a good response, and very often there are some residual chemoresistant leukemic cells that may survive. Detecting these surviving leukemia cells is very hard, and over the years we have become better and better. So for example, when PCR was starting to be used, we were able to see much more minimal residual disease than before we were able to have these molecular methods. Then the next question is, how are these chemoresistant cells that survive the first round of treatment are eventually reactivated and develop into relapse again? And here is where we actually still have an incredible amount of unknowns. And for example, it is very useful for us to team up with researchers that work with completely different types of cancers. For example, researchers that work with breast cancer, because it metastasizes to the bone, and we know that there are cells that are able to initiate breast cancer metastasis in the bone and yet remain incredibly quiescent for many years, sometimes throughout the life of the person, or sometimes become activated and will develop into the metastasis. And I think something similar is happening also for leukemia patients. We just don't really understand all the mechanisms behind this. There are some telltales. For example, the clinicians tell me that if after chemotherapy, once there is remission, patients recover blood count well, they have a lower chance of having relapse develop, while if the healthy blood counts never recover so well, there is a higher chance for relapse to develop. And this to me really speaks about, after chemotherapy, having healthy hematopoiesis recover, the stem cells and the progenitor cells generating the cells that are needed, and really tilting the balance in favor of healthy hematopoiesis and against the malignant hematopoiesis. And that's why I really very much see the bone marrow as a battleground when this happens.
All right. Thank you very much for this. Maybe as a follow-up about the therapies available, Kenneth is asking, what do you think about CAR T cells?
So CAR T cells are one type of cells that are used for immunotherapy. And I feel like I have to say obviously, but we all know that there is a lot of enthusiasm for the use of immunotherapy. And this really is possibly the most promising approach in cancer and leukemia treatment that we've seen over the last number of years. So yes, I'm absolutely enthusiastic about these cells. Things that we need to keep in mind is that we know that there are some challenges, specifically when we are looking, for example, at acute myeloid leukemia, which is the leukemia that we are deeply studying at the moment. There are a number of challenges. One of them is that the CAR T cells work because they recognize antigens that make the tumor cells different from the healthy cells. And in the case of acute myeloid leukemia, unfortunately, the leukemic cells look very, very similar to the healthy cells. And so it is very hard to identify an antigen that will drive the T cell to kill specifically the leukemia, but not the healthy stem cells. But of course, a number of groups are working on this, and I'm sure breakthroughs will come. We also need the CAR T cells to do well within the bone marrow and remain active. And for example, one of the things we know is that several cancers, including leukemia, are able to inactivate immune responses. And so very, very recently, we're in the process now of looking at the interactions between, for example, AML cells and T cells, trying to understand what it is that makes a T cell win against AML, or what it is that makes an AML cell win against the T cells, which means the T cell is no longer active and no longer working. So my short answer is I'm incredibly enthusiastic. I really think it is the future. And the longer answer is we need to learn a lot more about it so that it really can become a wonderful future.
So I'm curious about this, to what extent immunotherapy will be driven by CRISPR-Cas9. Is that a key technology in this?
CRISPR-Cas9 has been incredibly useful, and it is used in many fields, and I think we will see it used more and more in future. Whether it has to be used in order to generate the CAR T cells that we need, I don't know. I actually don't generate CAR T cells myself, so I don't know the details of this. But it's so based on my own experience, which is really not generating CAR T cells, not actually using CRISPR-Cas9. I'm curious myself to see what the future will bring. But as a technique, CRISPR-Cas9, again, it has been one of these absolutely revolutionizing discoveries that is making everything possible.
Thank you. So here's an anonymous question. What do you think would happen to an individual who has gone through a severe infection and subsequently gets leukemia? Would their survival or outcome be significantly worse because of the damage already caused by the infection? And also vice versa. Do you know of if osteoblasts come back after leukemia?
Yes, so I'll take the second question first, because I know the answer a little bit better, which is that yes, they do come back. What is interesting to understand is to what extent do they come back, and also whether not only the osteoblasts but also hematopoietic cells may have a memory of the leukemia having been there, and potentially also the chemotherapy having been there. And these are things that we don't understand yet. And this is why, in answer to the first question, what exactly are the relationships and what are the chances of for example, having infection and then leukemia, then recovering better or recovering worse, I really don't have the answer, because we don't know enough in order to have the answer. But I hope that in a number of years we will do.
Thank you. Here's another question. How comparable is the data that your research group are able to generate from mice to what we would expect to see in humans?
This is a very interesting question, and it is something that we ask ourselves all the time, and it is one of the reasons why we like to work alongside clinicians that keep reminding us of what it is that happens to patients and what it is that is more relevant to find. So far, the things that we have checked actually were consistent. So for example, we were able to put there are systems that allow you to put human leukemia cells into mice that are permissive to their growth. And in the case of T-ALL cells, we could see them being migratory and being more migratory following chemotherapy. We could see loss of osteoblasts in bone marrow sections of T-ALL patients. We could see loss of endosteal vessels in bone marrow sections from AML patients. So far, we have had a fairly high correlation between what we see in mouse and what we see in humans. But I will probably never stress enough that whatever we see in the mouse, it is very important that we go test whether it's relevant for humans as well before we take it further.
All right, thank you for this. So, Azin Khan asks, why would antifolate used by Professor Faber show good results, but later on the cancer always comes back?
So this sort of goes back to what we were discussing earlier about targeted therapy and how does it work, will it be the answer and everything. I think that the simplest way to look at this is that when we're looking at cancer and we are in a cancer therapy, what we are seeing is almost we are seeing evolution happening very quickly and the survival of the fittest in a way. So already as the cancer first developed, it means that there are some cells that are the malignant cells that are able to grow much more effectively than the healthy cells, for example, in the bone marrow, such as in the images that I have shown here. And then when we apply chemotherapy, chemotherapy will cause a huge pressure for some cancer cells to survive. And the cells that survive typically are cells that are a little bit different from the bulk of the tumor that we were treating initially, and they have something that allows them to survive. What the something is again incredibly complex. Sometimes the cells will develop new mutations that will make the receptor for the chemotherapy no longer sensitive to it. For example, further mutations in BCR-ABL that have been identified. Sometimes they manage to lodge themselves in a microenvironment that protects them. And we think this is the case, for example, it can be the case for AML cells in the situation where we have lost the endosteal vessels. We end up having an area close to the endosteum where cells are relatively far from the circulation, and chemotherapy is delivered through the circulation. So you can imagine that if you're further away from it, chances are you may be getting a little bit less chemotherapy and survive a little bit better. And this is just a hypothesis we have, and we have not really tested, but it's a potential mechanism that we have out there. We also know a number of groups have discovered very recently, and this is something that everybody's incredibly curious about. We have discovered that not only leukemia cells, but also healthy cells interact with the neighboring cells, meaning they can have protrusions and touch each other in a way, but they can actually have very deep interactions, and they can generate, at least in vitro, we call them nanotubes, which are literally long arms in which two cells can communicate, and they can exchange material from the cytosol, so from the inside of a cell to the inside of the other cell. And for example, mitochondria can be exchanged in this way. Mitochondria are the lungs of cells, and they are some of the cellular components that are very heavily affected by chemotherapy. And it has been shown, for example, for B-ALL, that the leukemia cells can acquire healthy mitochondria from the stroma and therefore become more resistant to the chemotherapy because they can now get new healthy lungs in a way. We can look at it that way. So the mechanisms are incredibly complex. They are different for each therapy, for each disease, and that's why there is so much effort put into understanding them.
All right, thank you. Leona Ahmed is asking, one of the emerging ways to treat leukemia is by using stem cells. After these stem cells have been injected into the bone marrow, how do these cells know to differentiate into healthy blood cells as opposed to another cell which are needed in the blood, in the body?
Yes, so this is a question that is particularly dear to me, because the reason why bone marrow transplantation works as a therapy in the clinic is because it basically is transplantation of hematopoietic stem cells. Now, hematopoietic stem cells are very special stem cells compared to stem cells from other tissues. So for example, in most tissues, stem cells will be in specific areas, and there they are, there we find them. Hematopoietic stem cells are in the bone marrow, but they have this funny characteristic whereby at any one time, if we look in the peripheral blood of an animal or of a person, a very small proportion of hematopoietic stem cells is in the blood and is not in the bone marrow. And it has been calculated that it quickly goes back to the bone marrow, but the reason why the stem cells go into the blood, no one quite knows. This was a small digression, mostly to say that the hematopoietic stem cells are good at leaving the bone marrow and going back to it, and their ability to go back to it is what we take, when I say we again, clinicians take advantage of when performing bone marrow transplantation or hematopoietic stem cell transplantation, because the bone marrow cells or hematopoietic stem cells are simply infused intravenously into the recipient, and they know to go to the bone marrow. We were able to do experiment in mice where we could calculate how long it would take for a hematopoietic stem cells injected into the blood to go to the marrow, and it's incredible. It takes minutes. I was able to look at situations in which bone marrow transplantation doesn't work so well, and we found that in a number of cases that the issue was that those mutant stem cells were not able to go to the bone marrow. So in answer to the question, how do the HSCs go to the bone marrow and reconstitute hematopoiesis there, is because they're really programmed to go to the bone marrow and to generate hematopoietic cells. Then how does this treat leukemia? Is because as a result of regenerating hematopoiesis, the transplanted stem cells will also regenerate the immune system. And this immune system is an immune system that has never seen the host nor the leukemia. So the reason why I mentioned these two things is because the bone marrow transplantation can have some bad side effects, which are what we call graft versus host disease or reaction, in which the immune cells start reacting against various parts of the body of the patient, and this can be managed by using immunosuppressive therapy. But the excellent outcome of hematopoietic stem cell transplantation is what we call graft versus leukemia reaction. And here we are back to the discussion about immunotherapies and CAR T cells. And literally, bone marrow transplantation, we now know it was the very first case of immunotherapy used in the clinic, because the T cells generated by the hematopoietic stem cells can recognize the leukemia and can actually entirely get rid of it. So that's how it goes.
Thank you. And maybe a question I could have used as a follow-up earlier on. Do you know, sorry, this is an anonymous question. Do you know how the current COVID crisis is going to affect leukemias?
I don't, other than I hope so. There's of course a number of studies. So in the long term, I think from current COVID patients, there are a number of studies taking place whereby samples are collected, and I think we're going to learn a huge lot about what happens to the hematopoietic system of these patients, and we will see over the years whether the incidence of other diseases, including hematological cancer, will be different in these patients, especially in relation to how severe COVID was. Then in terms of actual cancer treatment, and I think that very much depends on how the hospitals are organized. The clinicians I work with here have been telling me that they have been working in fairly large hospitals where they have been able to keep the hematological cancer patients completely separate. And so, for example, they have been able to keep performing bone marrow transplantation even during the pandemic, because the bone marrow transplantation ward was so isolated from everywhere else and everything was tested so incredibly well that they could keep it going. And the reason why they kept so isolated is because in these wards we have incredibly immunocompromised patients, because we need to eliminate the immune system of these patients before having it reconstituted by the new cells. And so we have a few weeks which are incredibly critical, but it looks like it has been possible to achieve it. So hopefully the impact has not been too drastic.
All right.
But I'm not in the hospitals myself, so I will not be able to say anything more precise.
Thank you. Another question, an anonymous question: can blood cancers metastasize on the brain?
It can, actually. And this is the case especially for T-ALL cells, for example. And this is a reason why chemotherapy for T-ALL patients is given intravenously, but often is given intrathecally. So it is given, so it needs to pass the blood-brain barrier, and so it needs to be delivered beyond the blood-brain barrier. And actually, I have been fortunate to be involved in a study very recently with a colleague here at Imperial, where they are studying the effect of the anesthesia that is used together with the intrathecal delivery of chemotherapy, because it seems that specific types of anesthesia may have an effect on the leukemia itself, with some types of anesthetic having this effect of making the leukemia more susceptible to the chemotherapy. And so, yes, so in answer to the question, yes, this does happen. And it needs a completely different treatment because of the blood-brain barrier. But again, breakthroughs are coming.
Thank you. Here's a question that is maybe also close to how we started collaborating. Anonymous says, really cool images. How long does one of these experiments typically take, and how hard is it to extract data from these pictures?
How long the experiment takes depends a little bit on many parameters, but many of the images I have shown of the bird's eye view of the bone marrow contained within the frontal bones takes us, I would say, between, when we try to do it fast, we can do it in about an hour, I think, hour and a half. depending on the resolution that we use. But some of the images I've shown you were acquired within about an hour each. So we can go relatively fast. When we're looking at larger areas, so for example, we want to go deeper in the tissue, or we want to really acquire a great amount of detail, and for example, the image of the whole femur that I showed you earlier, I think that one took about 3 days to acquire. And this is just for the acquisition. In terms of data analysis, it is challenging. And at the moment, actually, I think the bottleneck of all the research we do is not so much anymore in generating the images, but it is in actually extracting the numbers from the images and making sure that the numbers we extract are meaningful and the data extraction is done in the proper way. And then I think where we can go fast again is the statistical analysis of the data and working with modelers and to interpret the data and generate new hypotheses. But what I see at the moment, a big bottleneck is in the image analysis, because it requires people that are very good programmers and that are very interested in the biology, and yeah, it's just challenging. It's just not a turnkey thing. A similar challenge is seen by people that generate transcriptomics, for example, and single-cell RNA-seq. But my understanding from the community is that type of data is a lot more standardized, and so there is a lot more, oh, if so-and-so has done X, I will apply to and see what happens, and chances are it will be fine. But when we're looking at images, each project has its own type of images, and there's almost no two images the same. And so every time you kind of have to reinvent how to get the numbers out. So it is a great deal of challenge.
Yeah,
machine learning is starting to be helpful. So thankfully, we have artificial intelligence.
This has to be the last question now. What is the role of inflammation on infection and infection on leukemia? Sorry, the role of inflammation on infection and leukemia.
Again, it is a very good question. Something that has been seen, to go back again to the immunotherapies and why they can be so challenging, especially in the case of leukemia and acute myeloid leukemia, is that we know that when T cells are reacting against the leukemia, they generate cytokines, including, for example, interferon gamma. And interferon gamma actually leads to the upregulation on the receiving cells of a molecule which is called PD-L1. And this PD-L1 is actually one of the main mediators of immune suppression. And so this is an example in which we have an inflammation that should be good, but ends up being bad because it can end up silencing the T cells themselves. There is really a lot that we still need to learn about the relationship between the two. So actually, this is a lovely last question, because it really remains as a question, and I think in the next number of years we will start addressing it.
All right, many thanks for this, Cristina. It was really interesting. I'm afraid this is all we have time for today. I would like to thank everyone who has sent in a question, and I would like to apologize to every question I didn't get around asking Cristina. Also, many thanks for the audience and everyone who participated in the event. Of course, special thanks again to Professor Lo Celso for giving us an opportunity to enjoy her research. It was a fascinating talk on a very important subject. Thank you also for addressing the many questions. I suppose everybody agrees that this was a very important, very exciting subject area. So thank you very much, Cristina. To wrap up, I would like to encourage everybody to join us for the next Imperial Science Break event, which will take place on Thursday, the 5th of November, when the Food Security Group will be discussing the role of natural ecosystems and biodiversity in agriculture and food security. Thank you very much for joining us today, and thank you very much, Professor Lo Celso, for your wonderful talk. I look forward to seeing everybody soon. Goodbye for now.